Dear students, our lecture today will be about biochemistry of blood. Human blood as a fluid connective tissue has multiple essential body functions. First of all, this is transport function because blood transports gases, oxygen, CO2, different nutrients, hormones, and compounds that can be named as a metabolic waste. Thermal regulation, this is the next function of blood, pH regulation, and protection against blood loss and different pathogenic cells that can be in organism. Blood constituents. First of all, I want to see that this slide illustrates which components are present in blood, but we will discuss with you chemical compounds that are present in blood and functions of these components. According to this slide, you can find that approximately 40% these are proteins, 50% this is water, and another chemical compounds is in approximately 1%. Another components, these are cells that necessary for realization of function of blood. And we will discuss with you first of all proteins, and here you can find they are albumins, globulins, fibrinogen. You should understand the difference between serum and plasma, because sometimes we can say about serum proteins, sometimes about plasma chemical compounds. This is illustration. The difference, really, there is the difference between serum and plasma, and we can find approximately 15 differences between these two liquids, maybe more, but first of all, I want to say in chemical composition, in serum we can find proteins, electrolytes, antibodies, antigens, and hormones. In plasma, we can find different blood cells, and again proteins and chemical compounds. In serum, we can find less water, approximately 90%. In serum, we cannot find fibrinogen, and sometimes we can name serum as this is the plasma without fibrinogen. What about chemical composition of blood more? First of all, you should know all chemical compounds that present in blood can be divided on some groups. First of all, we can name organic substances and inorganic substances. Organic substances can be divided on two subgroups too. First of all, first group is proteins. Second group is non-protein organic compounds. Non-protein organic compounds can be divided on two subgroups too. Nitrogen-containing compounds and compounds they don't have nitrogen, non-nitrogen containing compound. And water from 90 to 91%. This quantity of water is present in our blood. First of all, about proteins, because proteins are very important molecule, and proteins can realize blood functions, and proteins have such functions as osmotic function, buffer function, blood clotting, blood viscosity, and protein regulate capillary permeability. Proteins are necessary for realization of transport function of different compounds, and if, for example, there is deficiency of lipids and carbohydrates, proteins of blood can be used by tissues, because blood proteins can be used as a sources of amino acids for our tissues. Plasma proteins you can see are albumins, globulins, and fibrinogen. Today we will discuss with you albumins and globulins. Here you can find that in serum we can find albumins and 4 fractions of globulins: alpha 1 globulins, alpha 2 globulins, beta globulins, and gamma globulins. And here you can find information about concentration of proteins. Total concentration of protein in blood is 65 to 85 gram per liter. And approximately 50% of all proteins, these are albumins. Concentration of albumins from 35 to 50 gram per liter. And albumins really are very important proteins because albumins have more functions than separated fractions of globulins. What does mean alpha 1, alpha 2, beta, and gamma globulins? These fractions we can analyze according to electrophoresis. If you remember, we discussed with you electrophoresis of blood serum proteins, and we named 5 fractions that can be taken from this electrophoresis. Paper electrophoresis of blood serum proteins can give 5 fractions, and these fractions are here. They are albumins, globulins, alpha 1 globulins, alpha 2 globulins, beta globulins, and gamma globulins. This picture illustrates the result of electrophoresis. We have discussed with you this picture. You can find here, this is a result of paper electrophoresis, and there are some fractions. The biggest fraction is albumin, because concentration of albumins in blood is higher, more than in other fractions. And we can calculate, not we can calculate, the densitometer can calculate concentration of albumins according to results of electrophoresis. Electrophoregram gives this information. You should know this picture. You should know normal electrophoregram, because as you remember, we discussed in the case of different diseases, electrophoresis of blood serum proteins can be used an additional method for diagnostic, because fractions of proteins can be changed according to pathology. We named 5 fractions in serum proteins: albumins, alpha 1, alpha 2 globulins, beta globulins, and gamma globulins. Practically every of these fractions has specific proteins. And this table illustrates which proteins are in every fraction. First of all, I want to say about albumins. In fraction of albumins, we can find albumin and prealbumin. As a rule, albumins are necessary for transport of different compounds, organic and inorganic compounds. Albumins participates in realization of buffer function of our blood, because there is a protein buffer system. Albumins is necessary for maintenance oncotic pressure, and albumins, by the way, can be used as a sources of amino acids for tissues if there is deficiency of energy or if there is deficiency of amino acids. In alpha 1 globulins we can find TBG, tyrosine binding globulin. According to the name, you understand this protein is necessary for transport of tyrosine. Transcartine is the next protein. Alpha 1 acidic glycoprotein, alpha 1 antitrypsin, and another proteins can be in this fraction. We will study with you all fractions, all these proteins and all functions of these proteins. Alpha-2 globulins. Here we can find haptoglobin, for example, macroglobulin, ceruloplasmin. All these proteins have specific functions. I'll say more about this later. Beta globulins again, we can find transferrin, hemopexin, lipoproteins, LDH, low density lipoproteins, and another proteins. I want to say that you should know functions for all these proteins, not only for beta and for albumins, for alpha-1, alpha-2 globulins, and gamma globulins. Gamma globulins are immunoglobulins. I hope you know that there are immunoglobulin G, M, A, D, E. And these proteins are important for realization of immune function of our blood. This slide illustrates electrophoregram of blood serum proteins, and in short form, which proteins are in which fraction of serum of blood. First of all, I want to say again about alpha-1 globulins. You should know which proteins are in this fraction. By the way, here there is mistake. You remember I said thyroxine binding globulin, TBG. In alpha-1, alpha-2 fraction, which proteins are present, and in fraction beta and immunoglobulins. A similar picture can be on the exam, and you should know which proteins are in which fractions, and you should know functions of these proteins. This slide illustrates functions of named proteins. For example, alpha globulins fraction alpha-1, alpha-2, you can find here. These fractions have haptoglobin, and here you can find functions of haptoglobin, ceruloplasmin, prothrombin, and another proteins. In the same time, you can find here functions of transferrin and another proteins. We will discuss functions of these proteins on our practical class. Some words about ratio, albumin to globulin, AG ratio. It is important because of clinical importance of this ratio. Normal range is 1.5 to 2.5, and you can find here the formula how this ratio can be calculated. If there is changing of this ratio, this pathological state is called as a dysproteinemia. Dysproteinemia can be as a result of different change in concentration of albumins and as a result of abnormal production of globulins. Low AG ratio can be as overproduction of globulins. It is logically according to this ratio. For example, in the case of myeloma or autoimmune diseases, or underproduction of albumins, for example, in the case of cirrhosis. Higher AD ratio can be in the case of underproduction of immunoglobulins. Maybe as a result of some genetic deficiencies or in some leukemias. I want to say again about total concentration of protein in blood plasma, because you must know this number. The total concentration of protein in blood plasma is 65 to 85 grams per liter. If concentration of blood protein will be low, in this case we can say about hyperproteinemia. Not only dysproteinemia can be. There is hyperproteinemia. There is hyperproteinemia. And in this case, we can say about a different level of proteins in blood. Hypoproteinemia, this is a low concentration, less than 65 gram per liter. As a rule, there are two main causes. First of all, maybe this is kidney disease, because in this case, organism will lose proteins, or liver disease, because in this case liver cannot synthesize enough quantity of proteins for blood, because practically all proteins of our blood are synthesized in liver. If we will say about albumins, albumins are synthesized in liver. Alpha 1, alpha 2 and part beta globulins are synthesized in liver. Only part of beta globulins and gamma globulins are synthesized in cells of immune system. And that is why hyperproteinemia can be a result of liver diseases. But I want to say that protein synthesis function of liver is very strong, and only if liver is very sick, only in this case. If really hepatocytes cannot synthesize proteins, we can find hyperproteinemia. First of all, we should say about kidney disease if you will find hyperproteinemia plus liver disease too. Hyperproteinemia can be as a result of protein deficiency in diet, because you remember there are essential amino acids, and deficiency especially of these amino acids can give hyperproteinemia, because in this case liver cannot synthesize proteins. Hyperproteinemia can be as a result of deficiency of water loss, or can be really in the case of overproduction of proteins. And that is why we can name relative and absolute hyperproteinemia. And in the case of hyperproteinemia, we can say about relative hyperproteinemia too, because really if water, there is overhydration, hyperhydration of organism, in this case we can find relative hyperproteinemia. And dysproteinemia, in this case, if you remember, I said about changing in protein GA ratio. This ratio can be changed, and changing of GA ratio can be according to different causes. Maybe hereditary diseases can be, maybe loss of certain protein functions, maybe proteolysis, proteosynthesis, diseases can give dysproteinemia. Again, it can be a result of dehydration, maybe inflammatory reactions can give dysproteinemia. As a result, we can find a lot of different causes for dysproteinemia, and GA ratio can give information about this pathological state. Next, I want to explain again in short form all about non-protein nitrogen-containing blood substances. These substances, nitrogen-containing compounds, can be named as a residual nitrogen or rest nitrogen, residual nitrogen. Why? Because concentration of these compounds can be defined after precipitation of proteins. Only after precipitation of proteins in supernatant we can define the concentration of chemical compounds that can be in this fraction. In a fraction of residual nitrogen we can find urea, approximately 50%, amino acids, uric acid, creatine, creatinine, bilirubin, indican, choline, nucleotides, polypeptides. All these compounds, the nature of these compounds we will discuss in practical class. You should be ready to discuss what does mean urea, and in which cases concentration of urea in blood can be increased, and we will discuss what does mean BUN, because it is important for medical practice. Normal concentration of residual nitrogen is 14.2 to 28.4 millimole per liter. If concentration of residual nitrogen will be increased, we can say about azotemia. Next chemical compounds of our blood are enzymes. All enzymes that present in our blood we can define on two big groups. First of all, function plasma enzymes. Function plasma enzymes can be defined as the secretory enzymes because these enzymes are synthesized and secreted into the blood, because in blood these enzymes have specific functions. These are blood clotting enzyme, for example. These are, for example, serum cholinesterase. All these enzymes are present in our blood because there are substrates for this enzyme in our blood. These enzymes are synthesized in the liver, and if, for example, there is liver diseases, activity of this enzyme in blood can be decreased. As a rule, we know activity of enzymes in blood can be increased because some enzymes can come from tissues into blood. But if this enzyme is secretory enzyme, liver diseases can give decreasing activity of this enzyme, and we can find it for serum cholinesterase. And these enzymes are called as the functional plasma enzymes because really activity of these enzymes in blood is more than in tissues. Next, non-functional plasma enzymes. Non-functional plasma enzymes can be divided in two subgroups: excretory enzymes and indicator enzymes. Excretory enzymes you can find here: alkaline phosphatase, lysine aminopeptidase, because this enzyme can be present in bile and can be excreted. Indicatory enzymes, these are tissue enzymes. And here you can find examples of tissue enzymes: alanine aminotransferase, aspartate aminotransferase, creatine phosphokinase or creatine kinase, lactate dehydrogenase, and others. As a rule, these enzymes, non-functional plasma enzymes, are present in our blood not in higher concentration. In low, these enzymes have low activity in blood. These enzymes are synthesized in different organs, and as a rule, activity of these enzymes will be increased in the case of increasing permeability of membranes. And why? Because enzymes of cells, because these are cells enzymes, will come into blood. Indicator enzymes can be divided on two subgroups. Indicators enzymes can be organ-specific and non-organ-specific. Organ-specific enzymes are present only in specific organs, and in other organs these enzymes are absent. For example, transamidinase. Transamidinase is an enzyme that we can name as organ-specific for kidneys. Transamidinase, if you remember, is necessary for the synthesis of creatine. The highest activity of this enzyme we can find in kidneys. Really, low activity of this enzyme we can find in pancreas, but as a rule we can explain transamidinase as an organ-specific enzyme for kidneys. Increasing activity of this organ-specific enzyme can give information about organ localization of pathological process. If you remember, I said blood has buffer functions. Blood has buffer systems, and that is why blood can maintain a constant pH. According to knowledge from biochemical chemistry, you know there are buffer solutions. Buffer solutions are used for different experiments if pH in this experiment should be constant. For blood, it is very important because in our organism, if you remember, enzymes can be active only in optimal pH. pH of arterial blood is approximately from 7.35 to 7.45, and this pH should be constant in our blood. There are 4 buffer systems for maintenance of this pH. There are 4 buffer systems, as I said. Here you can find, first of all, bicarbonate buffer system. This is the most power buffer system in our blood. Phosphate buffer system. Hemoglobin buffer system, by the way, maybe we can give second place for hemoglobin buffer system, and protein buffer system. We will discuss how this buffer system can realize this function on our practical classes. Next, I want to explain specificity of erythrocyte metabolism. Erythrocytes or red blood cells really have specificities, have specific metabolism because these cells are without nucleus, ribosomes, and mitochondria. If a cell without mitochondria, you understand, in this cell, only anaerobic conditions, anaerobic metabolism can be realized. And that is why the main source of energy for erythrocytes, this is anaerobic glycolysis. It is logically because the main function of erythrocytes is oxygen transport. In next specificity, in erythrocytes, pentose phosphate pathway is a very powerful process of glucose oxidation. Why, I'll explain later. In erythrocytes, there is production of 2,3-biphosphoglyscerate because this compound is necessary for regulation of affinity of hemoglobin to oxygen. In erythrocytes, hemoglobin is protected from oxidation. Because in erythrocytes there is powerful production of NADPH, there is glutathione, and glutathione reductase should work, and next enzyme that can protect hemoglobin against oxidation, this is methemoglobin reductase. This slide illustrates specificities of metabolism in erythrocytes. First of all, glucose is the main source of energy, and glucose is catabolized in anaerobic conditions. And here you can find the illustration, schematical illustration of anaerobic glycolysis. Glucose is catabolized to lactate. In the same time, in erythrocytes, pentose phosphate pathway is very active. Pentose phosphate pathway is illustrated here. Glucose 6-phosphate, 2 pentose phosphates. A pentose phosphate pathway or hexose monophosphate shunt. If you remember, pentose phosphate pathway produces NADPH. NADPH is not used for energy production. First of all, NADPH in erythrocytes is necessary for regeneration of glutathione. GS8, this is glutathione. This is glutathione in reductive form, and that is why we can say that NADP8 is necessary for reduction of glutathione. Glutathione in reductive form is important for erythrocytes because glutathione is necessary to work in antioxidant system. Normal concentration of glutathione is necessary for decreasing concentration of reactive oxygen species. Reactive oxygen species that can be produced in erythrocytes, and these active molecules are produced in erythrocytes, can provoke peroxide oxidation of membrane, and this situation can lead to hemolysis of erythrocytes. And that is why if, for example, concentration of NADP8 will be decreased, like in the case of deficiency of glucose 6-phosphate dehydrogenase, we can find hemolytic anemia in organism of the patient. Next, glycolysis produces NADH, and NADPH2 is produced in erythrocytes. It is important for reduction of methemoglobin to hemoglobin. Methemoglobin has iron in 3+ form, and in such form hemoglobin cannot participate in transport of oxygen. It is necessary to have enough concentration of hemoglobin with iron in 2+ form. Only in this case hemoglobin can realize transport function and can transport oxygen. Next, today we will say about 2,3-diphosphoglycerate. 2,3-diphosphoglycerate is formed in erythrocyte from 1,3-diphosphoglycerate. This is intermediate of glycolysis, and 2,3-diphosphoglycerate is necessary for the regulation of affinity of hemoglobin to oxygen. And now about hemoglobin. Hemoglobin in our blood is used for transport as the oxygen, as the CO2. The most common type of hemoglobin in adult is hemoglobin A. Hemoglobin is oligoprotein. This protein has 4 subunits: 2 alpha subunits, 2 alpha globins, and 2 beta subunits, 2 beta globins. Here, this is the illustration of the structure of hemoglobin, and you can find alpha chain, beta chain, beta chain. Plus, there is iron in the structure of hem. If you remember, our blood is red because of hemoglobin present. Hemoglobin is chromoprotein. When iron will be bonded with oxygen, color of hemoglobin will be changed, and our blood, that is why, is red. Not only hemoglobin A1 is present in our blood. Really, this is the main hemoglobin of our blood, alpha 2, beta 2. But in our blood, there is hemoglobin A2, hemoglobin F, and we can say today more about hemoglobin A1C, because definition of concentration of this hemoglobin has medical importance. What about hemoglobin A2? You can find here there are 2 alpha, 2 sigma subunits. Hemoglobin F has 2 alpha and 2 gamma subunits. Hemoglobin F has high affinity to oxygen, and it is why it is important to have low concentration of this hemoglobin in adults' blood. What about hemoglobin A1C? This hemoglobin is called glycated hemoglobin. According to this slide, you can find this is hemoglobin A1 plus carbohydrate C plus glucose. Glucose is attached to alpha 2 beta 2 non-enzymatically, and concentration of this hemoglobin in blood depends on concentration of glucose. If concentration of glucose is higher, in this case, there is increasing concentration of this hemoglobin. First of all, about concentration of hemoglobin in blood. For adults males, this concentration is from 130-160 gram per liter. In gram per deciliter, you can find this number, 13.5, 16.5 gram per deciliter. For adult female, again, from 120-140 in gram per liter. In gram per deciliter, this is 12 to 14.5 gram per deciliter. You must know these numbers. Oxygenation of hemoglobin depends on, first of all, partial pressure of oxygen and CO2. It is important. It is important to know that oxygenation of hemoglobin and realization of this function depends on pH, because there is realization of Bohr effect. And oxygenation of hemoglobin depends on concentration of 2,3-diphosphoglycerate, because concentration of this compound can change again the affinity to oxygen. You should know that there are different forms of hemoglobin, and you should know these forms, and you cannot change the names of this form. Oxyhemoglobin, this is hemoglobin with oxygen. There is deoxyhemoglobin. There is carb hemoglobin. This is compounds of hemoglobin and carbon dioxide. Carboxyhemoglobin. This is combination of hemoglobin with CO, carboxyhemoglobin. Methemoglobin I explained. In this case, iron in the structure of hem is present in 3+ form. And this hemoglobin, as you remember, cannot bind oxygen and cannot transport this very important gas. I said that glycated hemoglobin in medicine diagnostic is very important. This form of hemoglobin, when hemoglobin A1 is connected with glucose. This is a chemically linked component because for formation of hemoglobin A1C, enzymes are not necessary, and if concentration of glucose will be more than 3.3 to 5.5 millimole per liter, we can say about increased quantity of hemoglobin A1C. Definition of concentration of hemoglobin A1C sometimes can be more important than definition of only glucose concentration in blood. Why? Illustration is here. First of all, hemoglobin A1, as I said, can react with glucose in red blood cells non-enzymatically, and there is formation of hemoglobin A1C. This concentration of this hemoglobin can give more information because red blood cells live approximately 100-120 days in our blood, and that is why this concentration of hemoglobin A1C can give information about concentration of glucose in the last 3 months. If concentration of glycated hemoglobin is high, in this case, we can say about high concentration of glucose during the last 3 months. Here you can find which concentration of hemoglobin A1C is normal. Normal is before 5.7%. which percent? This is according to the concentration of total hemoglobin. You know, male should have approximately 140, 160 gram per liter. And before 5% from this concentration, we can say about normal concentration of hemoglobin A1C. Between 5.7 and 6.5, we can say about higher risk of, for example, diabetes mellitus, because as a rule, this test is used for diagnostic of this disease. And if concentration is more than 6.5, and we can say about constant higher concentration of glucose in blood, and it can be, for example, in the case of diabetes mellitus. This is exactly. Thank you for your attention. I hope my lecture was interesting and will be useful for you.